Hello, everyone, and welcome to today's broadcast, Fighting Against Air, Physiologic Oxygen and Reproducibility, presented by Dr. Alicia Henn, Chief Scientific Officer, BioSpherix. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Henn. I will now turn the presentation over to her.
All right, well, thank you, Javier, and thank you to LabRoots for giving me this opportunity to speak with you all today. I'm going to get right into my presentation here, and please invite you again, please send me questions. I always love to talk about my favorite topic here, and let's get started. So I may be the only presenter today that's going to ask you to close your eyes to start off our talk. And I want you to close your eyes and imagine your own cell biology lab. Stand in the doorway and think about what you see, what you'd normally see there. You see chairs, you see tables, you see benches, you see equipment. And I'm going to ask you, what of all that do your cells actually need? Because I'm going to make the case, and here you can open up your eyes, but I'm going to make the case here that your cells don't need any of that. They don't need any part of your traditional cell culture lab. We're going to talk about today what cells actually do need. So what do they need? Cells don't need the chairs and tables and equipment. They need unbroken physiologic conditions. That's really what cells need. And part of what we do at BioSpherix is try to present that environment for cells wherever you need the cells to be. We present a cytocentric environment for them. So this is what cells usually see in a traditional cell culture lab. They see an incubator that's easy for people to use. It's easy to get your hands in there. It's easy to get your face in there. But we all know it's not actually the optimal environment for cells. It's easy for cells to get contaminated in there, and conditions are highly variable. They're not physiologic. Every time the door opens and shuts, room air mixes with incubator environment, which causes variations in the environment. And that door doesn't open and shut the same number of times on a Sunday as it does on a Monday. We all know that. So how can we make an environment that's better for cells? Well, BioSpherix, we make equipment that provides cytocentric incubation, in that the doors on the incubators open only to controlled cell handling space, so that the cells are kept in optimal conditions wherever you need them to be. It's like being able to put your hands right in the incubator and manipulate your cells without the cells ever having to come out into room air. Likewise, people-centric cell handling is open to the room. This is biological safety cabinet where the cells are at suboptimal conditions. They're at room air temperatures, which is hypothermic for the cells. They're at room air carbon dioxide levels, which is hypocapnic for the cells. And they're at room air oxygen levels. And I'm going to spend most of the rest of this talk talking about oxygen levels, but this is hyperoxic for the cells. And I've got a reference there to a paper there if you want to look into more detail about how this cell environment and the room air environment is interrupted by people actually just working in that space. If you want to see more information about that. A cytocentric cell handling is closed, meaning that we take any equipment that the cells need and we put it into that optimized cell space. So microscopes, centrifuges, cell sorters, whatever equipment you need for cell care or cell analysis, we can put into this enclosed and optimized environment for the cells. Whether it's huge pipetting robots like this large automated TCAN robot we enclosed, whether it's for bioprinting, bioreactors, you name it, we can enclose it into a space that's optimized for the cells. And in that cell space, there's no room air. It's just filtered, tanked, medical grade gases that are used to provide that atmosphere. So we can dial in the CO2, the O2, whatever conditions we want for those cells. We can also control particles. We can control a volatile organic compound fumes. We can control all these critical cell parameters to make sure that the environment is optimized for those particular cells. More broadly, the Xvivo system that we build for our customers is an entire cytocentric facility that can protect an entire process from start to finish, so that once your cells go in, they can stay in controlled, optimized conditions out of highly variable room air for whatever you need to do for those cells the entire time you've got them, whether they need to grow and differentiate, whether you need to get data out of them, or whether you need to get a cell product out of them. We can control the environment for all of that. Now, it doesn't have to be a huge Xvivo system like this either to provide better conditions for your cells. We also make smaller chambers that you can put into an existing incubator to optimize conditions for cells in an existing incubator as well. I'm going to talk a little bit more about these and show you some data that some of our customers have generated with this a little later. So the cytocentric approach, what that really means is really taking the cell's point of view. And in this case, I'm going to talk about how important oxygen is from the cell's point of view. It seems like a very fundamental thing, but it's not something that we talk about enough. We're going to talk about the best oxygen levels for cells, both in vitro and in vivo. Now, oxygen in vivo can really be broken down into two large categories of factors. Those are factors that are cell relevant, such as the cell type that you're studying, what those cells are doing, what kind of state they're in. And there's also site-relevant factors. I'm going to start with these site-relevant factors to talk first about oxygen in vivo. So oxygen in vivo is really all about the same 3 L's that you see in real estate: location, location, location. It's all physiology-driven. As soon as you breathe air in from room air oxygen is about 21% oxygen. The minute that enters your upper airways, it's already mixing with air that you're exhaling. So the oxygen level immediately drops. So even in your lungs, oxygen's down to about 10, 13% oxygen, not anywhere near what room air is. And oxygen levels go down from there. And each organ type can have very different oxygen ranges depending upon the physiology of that particular environment for the cells. Excuse me. But you'll notice that all of these oxygen levels are far lower than the room air where we work with cells in traditional cell culture. And oxygen in vivo is never any one value, because oxygen isn't present in just one level. It's always present in gradients, and it's always in flux, because oxygen is always being delivered and being consumed to different tissues at different rates. So you get these very tissue-specific oxygen ranges, and those oxygen levels will fluctuate. There's even one paper that claimed that they were measuring oxygen levels very close to the pericapillary wall outside the circulation. And they were able to see what they saw was oxygen fluxes as single red blood cells pass by that one spot. So oxygen is very difficult to sort of pin down to one specific number. It's always in ranges and it's always changing. Also, as you move away from the blood circulation, oxygen levels drop quickly in normal, healthy tissues. So you can have very low oxygen levels in normal, healthy tissues. And then there are special areas that are of particular interest right now to the research and clinical areas, such as the bone marrow niche for stem cells. And this area is kind of interesting because it has a different physiology. In this area, you have sort of this river delta, this sinusoidal blood flow, where blood vessels interconnect and the blood vessel wall thins out, and the blood vessels spread out so that blood actual flow rate slows down there, which means that more oxygen can get pulled from the blood supply in the area as it's passing through these areas, and the slower flow rate leads to lower oxygen levels in those areas. So it's very much physiology driven. And this balance of oxygen being delivered and oxygen being consumed, of course, is disrupted in pathologies where you might have issues with the vasculature. And one very well-known pathology, of course, is in tumors, where you don't have good blood vessel development for that tissue. And you can get hypoxic areas at the center of these tumors that can be extremely low in oxygen. There's also a very high competition rate for oxygen at those sites. So you get naturally very low oxygen levels. And then there's areas even very across small areas, such as in the B cell germinal center in the tissue, you can get different oxygen levels in different areas. Whereas like in the light zone, you can have low oxygen levels. In the dark zone, you can have high oxygen levels. And as the cells circulate between these different levels, between these different areas, they're subjected to the different oxygen levels. And they have also very different functions in these different zones, which really leads to a lot of very interesting questions that people are exploring now about the connections between different oxygen levels across very small scales in vivo and what that means for cell function. In the intestines, in normal intestines, of course, the high competition rate from the microbes in the intestines will cause a very low oxygen level on the luminal side of the intestines, whereas on the other side, you can have very high oxygen levels because you got good blood flow. So you can get a high oxygen gradient across those tissues in normal healthy tissues. In a disrupted pathologic state, such as inflammatory bowel syndrome, however, they've picked up signals that some of these cells might be experiencing some hypoxic stress, some as if they're not getting enough oxygen. So there's some very interesting pathology-related oxygen effects going on there as well. Wounds, of course, are also an area because you've got bad delivery of oxygen and high competition from perhaps microbes at that, where oxygen plays a critical role in being able to heal those wounds. There's a recent paper by Evan Bort called The Importance of Controlling In Vitro Oxygen Tension to Accurately Model In Vivo Neurophysiology, which was published quite recently in Neurotoxicology, in which he really lays out the case that if we're not using oxygen levels that are more reflective of the in vivo situation, that we're not looking in the right place for answers to our most pressing questions about neurophysiology. And here he shows on a scale the in vitro oxygen levels that these cells are being exposed to and how far away they are from normal physiologic levels in the brain and pathophysiologic oxygen levels in the brain. So you can see there's very much a compelling case to using the proper oxygen levels for cells and trying to mirror what's going on in vitro. Other pathological states may include ischemia reperfusion, or high altitude sickness, or remote ischemic conditioning. All of these are cases, of course, in which oxygen is absolutely a critical parameter that must be regulated in vitro. So I'm going to stop here for a minute and change gears and talk a little about words, talk about terminology, and talking about using relative oxygen reference frames when we're talking about oxygen as a state. The old terminology, traditional terminology, has been really formulated on using room air as the reference frame. So room air is called normoxia, and anything lower than room air would be called hypoxia, which would include all the physiologic ranges of oxygen, whether they're actually pathologic or whether they're physiologic. And that doesn't really give you much room for going hyperoxic in any sort of in vivo relevant frame. So when we start talking about physiologic oxygen, it leads to a lot of confusion, because now your normoxia, a physiologic normoxia, actually isn't at room air, it's actually much lower oxygen level. And anything that's hypoxic, you would want to be pathologic, but it can't be. And you want to be able to talk about hyperoxia, because too much oxygen is also stressful and damaging for cells too. So, but being able to use these, trying to reuse these normoxia, hypoxia terms in a physiologic frame as opposed to the room air frame makes it quite confusing. And we've seen actual cases where, you know, trying to use the same term hypoxia, whether it's physiologic or pathologic, has led to actually erroneous conclusions. For instance, you can take the same oxygen percentage-wise, say 5% oxygen, and we try to apply that across different states physiologically, and you can get either a pathologic hypoxia situation, such as 5% oxygen will be too low for perhaps an upper airway cell. It might be just right for a cell of capillary endothelium. It might be far too high for something like a bone cell or a chondrocyte that would normally be exposed to very low oxygen levels. So because different oxygen states really depend entirely upon the physiology, we encourage people to sort of switch reference frames when it comes to terminology, move away from this people-centric room air term normoxia, because there's nothing normal about room air for cells and tissues, and start using the term physioxia, which is more cytocentric because it really shifts the frame of reference to the physiologic state. So coming back to our oxygen relevant factors, let's talk about the cell relevant factors now. Talk about cell type, differentiation state, activation state, whether those cells are functioning to proliferate, to migrate, and how important it is to regulate oxygen for those. And here I'm going to talk a little about immunometabolism, because this is a new, exciting, vibrant field that's really sprung up quite recently, just in the last few years. Tying what we were all taught in our basic biochemistry classes about metabolism, and tying it in new ways to cell function in such a way that we're seeing that basic metabolic machinery does more than just support cell function, it really controls it. So what we were all taught was that cells, when they have abundant oxygen, tend to use oxidative phosphorylation, using oxygen as the final electron acceptor in metabolism. And when cells are deprived of oxygen, they'll switch to glycolysis. Excuse me. But what we're finding is that there are conditions where cells will use glycolysis even when oxygen levels are high. Even when they're given more oxygen, they don't seem to be using it switching to oxidative phosphorylation, but using glycolysis machinery instead, which is really interesting because in the past we've really thought about metabolism functioning mainly for energy production. But we know that for immunity, you need also effector reactive oxygen species production, and you need biomass production. And this biomass production may be why cells are choosing, particularly T cells and T lymphocytes, are using glycolysis rather than oxidative phosphorylation. So cells they seem to be using different metabolic pathways when they're in different states. So a naive lymphocyte may his needs are relatively low energy, low biosynthetic rate. They tend to use things more of pyruvate and more fatty acid oxidation and more oxidative phosphorylation. Okay, so cells at different states are using these different metabolic pathways. Once your resting lymphocytes starts dividing rapidly, then it seems to switch to glycolysis in order to support biomass production that it needs for the daughter cells. Likewise, hematopoietic stem cells, which we'd find in that niche site in bone marrow, also tend to use aerobic glycolysis. There was a seminal paper published in 2016 coming out of the Carl June lab about CAR T cells and how important metabolism is for the CAR T cells. And they found some very interesting things looking at cells that were using different CAR T molecules that had different co-stimulatory modules in them, finding that those that had 4-1BB co-stimulation had increased fatty acid oxidation, increased oxygen, increased proliferation, increased survival in vitro, and a different T cell phenotype in that they had more central memory than the ones that were using the CD28 CAR. And these cells tended to be more glycolytic and have more effector memory phenotype. Also, there's evidence out there that CTLs in culture, regular CTLs, cytolytic T cells, in higher oxygen culture tend to use more fatty acid oxidation and have more proliferation going on, where in lower oxygen, they tend to have more lytic activity and less proliferation going on. And also their cytokine profiles are different as well. So oxygen for T cell culture can be absolutely critical. We also have a recent collaboration with ProMab and ASEA that resulted in a wonderful poster being presented at the CAR T Summit just a week or two ago. And I'd be happy to share a link to that. If you're interested, please let me know. And they presented the latest data on using controlled oxygen environments for CAR T environmental optimization. So what do my cells need? Let's come back to this question. Again, I don't know what cell type each of you is working in, but I can tell you without a doubt that room air oxygen is irrelevant for your cell type at best. But we all have to work with the cells in vitro. So let's talk about oxygen in vitro. So in vitro oxygen comes from the incubator, and your room air incubator is not at 21% oxygen, which is a fact that doesn't get a lot of airplay, as well as something you might not be aware of. Because of the increased CO2, of course, that we're pumping into a room air incubator, and because of the increased humidity in a room air incubator, you can't have 21% oxygen in there, because 21% is what room air oxygen is in the completely dry outdoor sea level environment. So if you calculate it out, the most oxygen you can have in there is about 18.6%. When we actually went around and measured room air incubator oxygen in several different institutes across upstate New York, we found that it averaged about 17.2% when you first close the door over the first couple hours. Now, if you open and close the door on your incubator, that tends to perturb the oxygen level quite a bit, because when you open the door, the CO2 all rushes out of your incubator and oxygen from the room rushes in. And then when you close the door again, and we've all seen this, you close the door on your incubator and a little light comes on, says that CO2 is charging up. And we watch the CO2 go back up to 5%. But what we don't think about is what's happening to the oxygen level while that's happening. The oxygen is going out that little sponge in the side of the incubator in the back, because the air's got to go somewhere if the CO2 is charging up, right? So you can actually drive the oxygen level all over the place in your room air incubator just by opening and closing the door. So just putting one of these chambers inside of your incubator, one of our C chambers in there, will help control this variability that you see from the incubator door being open and shut. And then if you put multiple chambers in there, you can even have multiple environments in there for your cells that you know are well controlled. So oxygen in vitro comes from the incubator. It has to pass through several phases before it actually gets to your cells. There's the gas phase outside your cell culture vessel. There's your gas phase inside the headspace of your cell culture vessel. And then there's the liquid phase of your cell culture vessel that also has to go through. And oxygen is very poorly soluble in aqueous medium. So this isn't a trivial thing. The depth of the medium has a big effect on how long it takes that oxygen to get to your cells if they're laying on the bottom of the cell culture vessel. And how dense those cells are, or how fast your oxygen consumption rate is, also will vary as your cells expand. So we say that pericellular oxygen is completely protocol dependent, and that all of these factors go into what the actual oxygen level it is that your cells see in the incubator. Oxygen in vitro also comes from the refrigerator. And what I mean by that is that because oxygen is so poorly soluble in aqueous medium, it takes hours for it to equilibrate with the gas levels in the incubator. So it is really essential that if you're trying to control oxygen levels that your cells see, that you precondition medium or other liquids that your cells are going to be exposed to the proper oxygen level to reduce the time that they're out of optimum. You need to work fast and you need to work smoothly with them in the BSC to minimize mixing with room air while you're working with them. And doing that, you can be very successful in controlling the oxygen level that your cells see. But you have to know how to minimize the time that they're out of optimum. But they're only out of the incubator for a few minutes. I hear this all the time because oxygen in vitro comes from the room as well when you handle your cells. Why do we need full-time physiologic simulation for these, even for cell handling? Well, there was a paper published in 2015 by the Broxmeyer group, which showed this very, very clearly. They defined a term called extraphysiologic oxygen shock, where they showed that cells during isolation, when they're first taken out of the body, if they're exposed to room air for as little as 15 minutes, they can have a rapid deleterious effect on those cells and on their function. They're working with hematopoietic stem cells in that case. And it's because of this protein. We know a lot more about this than we used to. HIF1 alpha at room air, and cells that are being exposed to room air oxygen displays one of the shortest half-lives among cellular proteins. It's controlled entirely at the protein level, not the RNA level, not the DNA level. It's controlled at the protein level, which means that even short periods of time out of optimum can actually affect cells. I'm showing you this from one of the early papers on HIF1 alpha, and I just wanted to show you the time scale on these figures. It just takes, we're talking single minutes of here out of optimum for those cells to be affected. And downstream of HIF1 are absolutely critical cellular functions. We're talking about functions that affect metabolism, cell death, cell cycle, differentiation, and all of these cell signaling intermediates and growth factors are affected by different oxygen levels through the action of HIF1 alpha. So oxygen is fundamental, but it's complex. When oxygen levels fall for cells, that can increase HIF1 stabilization, which can increase your glycolytic enzymes, increase your glucose transporters. And in lymphocytes, as I mentioned before, that affects their phenotype, affects their function. It also affects the functioning of natural killer cells, or NK, which are becoming more and more important clinically. It also affects myeloid cell function, macrophage. There's also one phase of the cell cycle. It's not regulated by cyclin CDKs, rather is regulated by the redox state of the cell, which means that if you have a lot of reactive oxygen species present there because the cells are trying to deal with oxygen levels that are too high, that can fundamentally affect cell cycle no matter what the cell type. We have a collaborator at University of Rochester that has been doing some work with B cells, and they've been able to show that different oxygen levels using this chamber gives them very different B cell functions in culture. So it looks like cell culture oxygen is critical for B cell function as well, and also for cord blood HSC. And if oxygen levels are too high, it can very much be impair the function of hematopoietic progenitor cells, as shown by this paper by Roger Rohn recently. So I'm going to shift gears again here and talk, because we've talked about oxygen in vitro, we've talked about oxygen in vivo as one of these critical cell parameters that are so important for getting the best data out of our cells and getting data that's relevant out of our cells, relevant for the in vivo situation. I'm going to talk a little bit about how controlling these conditions is critical for better reproducibility. Because once we can control the environment wherever the cells need to be in the lab, then we can really set about improving cell fate. So in our conventional cell conditions, every time you bring those cells out of the incubator, your oxygen's going off, your CO2 is going off, the temperature is going off, the pH is going off, and then you put them back in the incubator and those cells have to recover after the medium is finally recovered to incubator state. In cytocentric, full-time optimal conditions, you don't have these cells being thrown off constantly in suboptimal conditions. They have full-time optimal conditions. And we can take these different modules from our ex vivo systems and combine them in different ways to put them around any type of process that you need to do with your cells. So whether you need cell instrument chamber to control oxygen in there, or incubators with a processing chamber, we have buffer chambers and laminar flow, all of these work together to create this constant optimal cytocentric cell conditions. And once you've set up those modules in the way that you need them for your process, you can do this early in the research stage and then reuse those same modules over and over as you build your cells, build your product toward preclinical and clinical studies. And in the end, you end up with a completely GMP compliant cell production system. This contrasts with the best traditional clean rooms that we've got. Even in the highest tech, most advanced clean rooms that we have, people are still using incubators and BSC, so you still have this same variability for cells because it is primarily a people-centric environment, not a cytocentric environment for those cells. With a cytocentric environment for your cells, you can control not only all the cell parameters, and you can lock down the best optimal conditions for your cells, but then you can take those conditions and you can put them anywhere in the world and clone them. You can have verifiably the same conditions for those cells, whether you're in San Diego, whether you're in Saudi Arabia, or whether you're on a ship at sea. You can have verifiably the same conditions for the cells. So while when we talk about the reproducibility crisis for cells, and we've talked a lot about factors like differences in reagents, we talk about factors like reporting problems, we talk about cell identity problems and bad use of statistics, there's one factor that goes into every single cell-based assay performed on the planet, and that is cell conditions. And that is something we need to get into the larger discussion on reproducibility. So taking the capability for full-time optimization and full-time monitoring, we can reduce the variability that you see from experiment to experiment, from day to day, from year to year, from user to user. We can take the ability of full-time physiologic simulation and use that to reduce the variability when from translating from in vitro to animal, from in vitro to human. And we can take that clonable environment, that modular expandable environment, and use it to reduce the variability we see from lab to lab, from location to location, and in transferring our results from research to preclinical to clinical GMP cell production. So I'd like to stop there and leave some time for questions. Please feel free to reach out to me with my email. Also, come find me on LinkedIn. I run the in vitro reproducibility group there. I'd love for you to join the discussion. Come join us there, see what we're talking about. And check out our blog, www.BioSpherix.com/Cytocentric. Thank you very much.
Thank you, Dr. Henn, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Now let's get started. Our first question is, cells have grown fine in room air incubators for decades. Why change now?
Well, that's a really interesting question. There have been some publications recently that have shown that cells that are grown in room air oxygen, even for decades, are still being affected by the higher oxygen levels there. Their GSH levels, the glutathione levels are off, their redox status is off, and they may give erroneous results because of this oxygen artifact that is imposed on them by being grown in room air oxygen. So even for cells that have been in culture for decades, it's still very important to use physiologic oxygen levels.
And if HIF levels change that fast in room air oxygen, how can I ever get good data if I work in a room air ESC?
Yeah, I get this question a lot because we know that oxygen levels can affect HIF levels very quickly, but because your oxygen is poorly soluble in cell culture medium, that gives you a little bit of leeway to be able to change that medium as long as you're using pre-equilibrated oxygen levels in your media. It gives you a better chance of not disrupting the HIF status of those cells if you work smoothly, work quickly in a BSC. And I know our customers have been able to do it with our chambers, so I know that you can do it. Of course, working in a completely enclosed and controlled cell handling space, like in our Xvivo, is going to give you absolute control, and you don't have to worry about those factors.
And I've seen papers that said that hypoxic culture didn't make a difference. Why should I go through the trouble to work in low oxygen?
Yeah, well, we didn't understand HIF. We didn't understand how quickly cells were regulated by oxygen changes before we really knew about HIF-1 alpha. So there have been a lot of studies out there done without the proper conditions for controlling oxygen well for cells that I think have given very confusing results and misleading results. And we're seeing more and more papers now that people have better equipment, they have better oxygen detection ability, and they have better processes for controlling oxygen for cells. We're getting better data that oxygen actually is absolutely critical for most cell types.
All right, and our last question. I don't have space for a big system like that in my lab. Do you have anything smaller that can allow me to control oxygen during cell handling?
Yes, we do. We have a full range of products that can control oxygen for you for both incubation and for cell handling for all different size spaces and all different budgets, and all different needs. So please contact us and we can guide you to the best technology that would fit your space and your budget.
All right, well, I would like to once again thank Dr. Henn for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.